Okay, so this is the third video in the fundamentals of biology, and today we will be going over the cell parts and the cell membrane. So let's go over the two basic types of cells. You have the eukaryotic and the prokaryotic. So the first main difference between them is a eukaryotic is the large size, whereas a prokaryotic will be your smaller cells. And in a eukaryotic cell, the DNA will be linear, whereas a prokaryotic will have a circular DNA. A eukaryotic will contain a nucleus as shown here. However, a prokaryotic will not have a nucleus, where the DNA would usually be stored in a eukaryotic cell. And the major difference between them is a eukaryotic cell will have a membrane-bound organelles, which is why you have things like animals and plants, and fungi having things like the nucleus, an endoplasmic reticulum, a Golgi apparatus, mitochondria, plastids, lysosomes, and vacuoles, and we'll go over those later. And yeah, in prokaryotic cells, there's no membrane-bound organelles, which means it will only have things such as ribosomes, cell walls, and a cytoskeleton. And a eukaryotic cell will usually reproduce through mitosis, whereas a prokaryotic cell will reproduce through binary fission, such as bacteria and archaebacteria. So the endomembrane system is basically a system of organelles that modify, package, and transport things such as lipids and proteins. And this can only occur within eukaryotic cells because the endomembrane system involves membrane-bound organelles. So first we have things like the rough ER, or the rough endoplasmic reticulum. It looks like a series of flat stacks as shown in this picture, and they are called rough because they contain ribosomes. So what are ribosomes anyways? Ribosomes are basically these red dots, and they produce proteins. And within the rough ER, proteins are modified within the lumen, or the hollow space within the ER. And whenever the ER produces vesicles with these lipids and proteins, they will send it to the Golgi apparatus. However, there's also a smooth ER, and the purpose of this is to detoxify poisons, store calcium ions, and create steroids. So after the vesicles are packaged and sent to the Golgi from the rough ER, uh, the the proteins inside these vesicles will be sorted, tagged, packaged, and distributed. So you can see that the Golgi apparatus is a series of flattened discs as well, similar to the rough ER. And the Golgi apparatus has a cis and a trans side. So basically, it will travel first from the ER, and this vesicle will first go to the cis face. And as it goes down, it will go down to the trans. Another major organelle within the endomembrane system is the lysosome. So what this does is it breaks down unused structures through the usage of enzymes. However, they also participate in phagocytosis, which engulfs pathogens and toxic materials. Related to this is the peroxisome, but they're actually different because peroxisome produces hydrogen peroxide as a byproduct as a result of breaking down its materials. And what it breaks down are amino acids and fatty acids. And finally, the vacuole within the cell is used to store water and waste. And there are vacuoles within animal and plant cells. However, the animal cell, you can see this vacuole is much smaller than a plant cell because a plant cell will have a central vacuole, whereas animal cells will usually have probably a few smaller vacuoles throughout the entire cell. So now we will talk about the cell membrane. So what is the cell membrane even composed of? The main component of a cell membrane is the phospholipid, and they are unique because they have a hydrophobic and a hydrophilic side. So if you remember from lipids, they they have fatty acid tails and they are nonpolar, which means they are hydrophobic and fear outside environments such as water. Whereas the phosphate group is hydrophilic, which means it loves water. So it will usually be exposed to the outside, where polar attracts polar. So that's why it forms a phospholipid bilayer, which you can see here in this diagram. You can see that the fatty acid tails will point inwards, whereas the hydrophilic heads will point outwards. Another part of the cell membrane are integral proteins, and these are usually embedded within the actual phospholipids, and they form a channel or that can allow ions and other small molecules to pass. So you can see its many jobs in this picture. However, some proteins don't actually go through the entire phospholipid bilayer, and these are called peripheral proteins. So basically, they're not embedded within the hydrophobic section, and therefore they're usually more loosely attached. Another part is the cholesterol, and this minimizes the effect of temperature on fluidity. So they are embedded within these phospholipids. And finally, we have carbohydrates. And things such as glycoproteins and glycolipids help in cell recognition. So the difference between these are a glycoprotein, they attach to a protein, like the integral proteins, whereas a glycolipid will attach well to a phospholipid, one of these. So now let's talk about cell permeability. So what is able to actually pass through these phospholipid bilayers? So if the substance is small and nonpolar, such as oxygen and carbon dioxide, it will be easily able to pass through the phospholipid bilayer. Because if you think about it, the inside of the phospholipid bilayer is nonpolar. So nonpolar, it will pass through easily. However, if it's large and nonpolar, such as carbon rings, it still is able to go across that membrane. However, it will be much slower. However, if it's small and polar, such as water, it will require a channel protein. And specifically for water, you're going to need something called an aquaporin. And finally, for large and polar substances, such as glucose and ions, they will also require a channel protein. The next thing we'll talk about is the extracellular membrane, or the ECM. This is found outside the actual bilayer here. So these are composed of collagen fibers, and these are proteins, and these are used for tissue strength. And embedded within these collagen fibers are proteoglycan, which are made of proteins and polysaccharides. And something called fibronectins will connect these integrins to the ECM. And that basically keeps the structure of the entire cell together. Now we will talk about the cell wall. They are usually found in plants for support and structure, and they are made of microfibrils, which itself is made of cellulose. So now let's talk about the difference between passive transport and active transport. So the main difference is passive transport is able to occur without the use of energy, whereas active transport will. So diffusion is the movement of particles from a high to low concentration. So in this diagram, you can see originally, if you just put a lot of particles in here, they would like to spread out, so the concentration will become lower. And osmosis is basically the same idea as this, except it's with water. So as you can see here in this diagram, the right side will have less water, whereas the left side will have more water. So as a result, the water will move from high to low, and that's why there will be more water on the right side to balance out the large amount of particles in the original beaker. And finally, facilitated diffusion requires the use of a channel or carrier proteins, but it will still happen without energy because it will be usually from a high to low concentration. And some examples of active transport. Endocytosis and exocytosis is the movement of large substances either into or out of the cell. So as you can see, endocytosis is just the bringing in of particles into the cell. So phagocytosis is the solid particles going inside the cell, whereas pinocytosis is the use of liquid inside the cell. And then exocytosis is just the bringing out of large substances through vesicles, as we have shown earlier. The sodium potassium pump establishes and maintains something called the membrane voltage using things such as electrogenic pumps. So you can see in this diagram when you add sodium into the pump, you're going to use ATP, and that's going to require energy. And as these sodium exit the system, it's going to take in potassium, and that's going to maintain a charge within the cell. And finally, cotransport uses an electrochemical gradient and is generated by active transport. So what this means is, so you're going to use these proton pumps, for example, and it's going to pump these H plus ions out of the cell. And since there's a large buildup, they're going to want to diffuse down, and that's where the cotransporter comes in. So as these diffuse down, the sucrose is able to also go along with it. So that's how there can be a large buildup of sucrose, even though you would think that would have to go out. So now we will talk about what happens when cells are in different environments. So when it's in an isotonic solution, there will be equal salinity inside the environment and the cell. So you can see in a animal cell and a plant cell, there will be equal water movement inside and out. And this is going to happen because there will be no net osmosis happening. However, in a hypotonic solution, the environment will have a lower salinity, and as a result, water will start coming into the cell due to osmosis. And when this happens in an animal cell, the cell will usually burst and lyse. However, the plant cell will actually prefer this, and it will become turgid. And finally, if it's in a hypertonic environment, the environment will actually have the higher salinity. So as a result, the water will move out. And when this happens in an animal cell, such as a red blood cell, it will become shriveled, whereas in a plant cell, it will become shriveled as well, but it will be called plasmolyzed. And one final thing to talk about is why are cells actually so small? And the reason is for this is because they want to maximize their surface area to volume ratio. The reason for this is because they want to have the maximum interaction with their environment. So if you just think about it, if we have a large cube over here, the surface area to the volume will actually be smaller than if we just had a small cube over here, where the surface to volume area would be much larger. And the cell will also do this with folds within the membrane, such as the cristae and the mitochondria. So thanks for watching. This video covered the cell parts and their functions. We also went over cell membrane and permeability, and finally, we talked about how cells will transport substances throughout.